Good evening from Tel Aviv University, or good afternoon, whatever part of the world that you're joining us from. Can you hear me? Eladi? Wonderful.
Yes.
Okay, it's a real pleasure to see, well, behind the screen. I know that you're all there. I can see the numbers. So my name is Karen Abraham. I'm from Tel Aviv University. I'm the Vice Dean at the Faculty of Medicine, and it's my real pleasure to be moderating this evening's webinar. This has been an absolutely unprecedented time in history. I don't think that anyone alive today has gone through the tragedies and the difficulties that we're seeing worldwide. There's deaths, there's illnesses, there's economic disasters, many things that we may not recover from for a very long time. But this period, I think, in the past year has also given me and many others remarkable faith in humankind. We've watched entire societies band together. Health professionals work in a way maybe that they haven't done so together before. We become aware of the importance of public health, and we've had scientists from all over the world work 24/7 despite all of the difficulties of working under the lockdown conditions to figure out what's happening on a scientific level so that we could advance the research, not only for our biological and scientific understanding, but so that we can get a vaccine that's already started, certainly in Israel and in other places in the world. Here at Tel Aviv University, we've also started a Center for Combating Pandemics, which is directed by Professor Itai Ben-Har. And so we too have really joined the effort to try to make an impact, not only for those of us in our community and in Israel, but really all over the world. And I think that without a doubt, I can say that our two guests tonight who are joining us are part of that really enormous effort that's been undertaken. So the first, I'm going to introduce you to Dr. Ella Sklan, and let me give a brief introduction about both of them, and then we'll give them a chance to speak and a chance for questions and answers. And I do want to encourage you to already, during the time that they're speaking, to already put questions in the question and answer. You won't be able to ask your questions verbally. You'll have to do those all through me. So Ella, Dr. Ella Sklan did her PhD at Hebrew University in Jerusalem, and then did her postdoctoral fellowship at Stanford University. In 2009, she joined the Department of Clinical Microbiology and Immunology here at the Faculty of Medicine at Tel Aviv University. Her main research topic is on positive strand RNA viruses, on Zika viruses, dengue, hepatitis, and now SARS-CoV-2. And I remember Ella so well because last March, when we were just getting a hint that things were going bad, Ella volunteered to give a lecture to explain to the rest of the community who are not virologists what is really going on with this new virus. And we all went into a big hall at the Faculty of Medicine, and there were a few whispers here and there. Should we be going into this room together? We had no clue what was going on. Thankfully, there was no problems, even though we were all crowded into that room, a way that now when we look on TV and we see people collected in rooms like that, it seems bizarre to us. And since then, Ella has become famous, where she's on the news all the time. But really what's so special about the contribution that she's made is her sincerity and her seriousness in really explaining to the public and to us scientists who know less about this field, really what's happening. And today we're going to hear much more from her about vaccines. Before I let her start speaking, I want to introduce Dr. Adi Stern. So Adi is also a remarkable scientist in her own right. She did her PhD in the Faculty of Life Sciences with Professor Tal Pupko, and then she went on to do a postdoctoral fellowship at the Weizmann Institute of Science with Rotem Surek, some of you may know, was also a graduate of the Faculty of Medicine. They then went on to do a second postdoc at UCSF and at Berkeley. And in 2014, she joined the Shmuna School of Biomedicine and Cancer Research. And what she's been doing for many years is looking at the evolution and the genomics of viruses. Now, Adi also made the news very early on during this pandemic when she and her team published a paper showing where the greater proportions of the virus came from. And much to some people's chagrin, especially mine, because I'm from New Jersey, she showed that many of them came from the United States, and perhaps we should have closed our borders just a little earlier than we did. Since then, we've also heard a lot from Adi in the news, and has really made a difference in our ability to understand what's happening. So without further ado, Ella, I'd like to give you the stage. We'd like to hear, I understand you have a presentation you're going to show us. Give us a little bit of background. We'll then hear from Adi, and then I'd love to entertain everyone's questions.
So thank you very much, Professor Abraham, for the introduction. So we're going to talk about SARS-CoV vaccines. Somehow my presentation is not moving. Okay, so before we do that, let's first understand what is a vaccine. So when our body or our immune system is exposed to a foreign substance, it elicits an immune response. And you can see here on this graph, on the x-axis, the time from the exposure to that foreign substance, or as we like to call it, an antigen. And on the y-axis, you can see the antibody concentration, and immune response is more than just an antibody response, but we'll stick to that for now. So when we're exposed to the antigen, our immune system elicits a response. And as you can see here, upon our first exposure, this immune response will not be very high, and the antibody will not be very efficient. Fortunately for us, our immune system can, what we call, learn, and can improve its response on the next exposure. So when we will be exposed again to this antigen, we'll now have a much higher antibody response, and our antibodies will be much more efficient. And that's the basic idea behind vaccines, is to expose our immune system to these foreign substances or antigens. So when they will encounter the actual pathogen, they will elicit a stronger and a more efficient response. And those antigens that we expose our immune system to are usually parts of the pathogen. In order to better understand the various vaccine platforms I'm going to talk about, I need to refresh your minds on how information flows in our cells. So what you see here is a regular mammalian cells, one of the cells in our body. Most of you know our genetic material is DNA, as you can see here, and it's localized in the nucleus of our cell, which is stained here in purple. In our bodies, DNA it is the source of the information for production of a molecule called mRNA. And that molecule is exported outside of the nucleus to the cytoplasm of our cells, that area called the cytoplasm that you can see here. And in the cytoplasm, this mRNA molecule is translated into a protein. And proteins are the actual functional molecules in our cells. They do all the functions that are needed to be done in our cells. So DNA is made into RNA, and RNA is transported to outside of the nucleus, where it's translated to a protein. So vaccines are one of the amazing inventions of mankind. They saved the lives of millions of people over the years. And over the years, mankind has developed various platforms in order to produce vaccines. And now I'm going to go over some of these platforms, and then we can discuss which platforms are used in order to create vaccines for SARS-CoV-2, and I'll talk a little bit about the advantages of this and disadvantages of every platform. Before we do that, you can see here a scheme of SARS-CoV-2, of the virus. The genetic material of the virus is RNA, I showed you before. It skips the step of the DNA. Its genetic material is not DNA like ours, it's RNA. So when it goes into the cell, it can be directly translated into a protein. This RNA is encapsulated in a protein, and then it has a lipid envelope that you can see here. And on that envelope, the virus has several proteins. The most abundant protein is the protein that you can see here, called the spike protein. The role of this protein is to bind receptors on our cells, which helps them gain entry into our cells. Because it's the most abundant protein on the surface of the virus, that's basically what our immune system sees and what our immune system develops antibodies against, which is why this protein is used as the antigen in most of the vaccines that are developed, and I'm going to talk about it in a minute. So the first platform we're going to talk about is an inactivated vaccine. And I gave an example of each vaccine just below it. You can see here an example of inactivated vaccine is the vaccine for hepatitis A, which some of you might know, most of you probably already received. Basically, inactivation of the virus is the most simple way to go. You just take the virus, you inactivate it using various compounds, formaldehyde or something else, and then you inject it into the body. The advantage of this platform is that it's relatively simple. You just have to produce the virus. It's a disadvantage, for example, in the case of SARS-CoV-2, because it's a highly infectious virus and it's not that easy to handle if you want to make a large amount of it. Another disadvantage of this kind of vaccines is that sometimes during inactivation, you harm a little bit the antigens. And most of them don't elicit a strong response like the original. Some of them don't elicit a strong response like the original virus. In the context of SARS-CoV-2, some of the vaccines produced in China already approved there and already given to some of the population are inactivated vaccines. The next platform we're going to talk about is attenuated vaccines. Basically, some of the flu vaccines that we get through the nose mostly are attenuated vaccines. Some of the polio vaccines that some of you maybe remember were attenuated vaccines. Basically, what you do is you change the virus in a way that it is not going to cause disease anymore. There are a couple of ways to do it. Today, you can do it in the lab. You can generate mutations. In the past, we used to passage it on cells of a different organism, like on cells, on monkey cells. That way it will adapt to the monkey cells, and when we'll return it to human, it won't replicate as efficiently and it won't create disease. The advantages of this is that it goes into the body the same way as as the pathogen goes in. So it elicits an immune response during its whole way in. So if the pathogen comes into in the upper respiratory tract, it will create also an immune response in the upper respiratory tract, and I'll explain why it's important in a couple of minutes. The disadvantages is it's very rare. It happens very rarely, but if some of you remember from the polio vaccine, for example, 1 to a couple of million, it reverted back to its pathogenic state. So that's a serious disadvantage, and it's more difficult to prove the safety of such vaccine, which is why we don't see at this point an attenuated vaccine for SARS-CoV-2. The next type of vaccine is a protein subunit vaccine. Basically, what you do is, in case of SARS-CoV-2, you produce the spike protein outside of the body artificially. The advantage of that is that you get a large amount of this protein into the body, and if it's produced correctly, it can make a very strong immune response. The disadvantage is that sometimes it's more expensive to produce, and sometimes when you produce the protein outside of the body, it becomes some of the modification that occurs in the body don't occur when you produce it outside of the body. So sometimes it's a bit less immunogenic. An example for that kind of a vaccine is a hepatitis B virus vaccine that most of you also probably got. The next platform is a viral-like particle. An example for that is the papilloma vaccine, a little bit more difficult to produce. Basically, it looks like a virus, but it doesn't have the genetic material inside. It just presents the viral protein on a viral-like platform. I'm not aware also of a SARS-CoV-2 vaccines being produced in that using this platform. The next platform we're going to talk about is replication competent and incompetent vectors. Basically, what you do is you take a virus that usually doesn't cause a serious disease or doesn't cause at all disease in humans, and you incorporate into its genetic material the information that codes for your protein of interest. In this case, the spike protein. An example for that is the recently approved Ebola vaccine. It's based on a virus called vesicular stomatitis virus. that expresses some of the Ebola proteins. And I put here a small flag of the state of Israel because the Israeli vaccine is also based on the same, just to remind me to talk about it, it's also based on the same platform, just like Ebola. It's a vesicular stomatitis virus, a virus that doesn't cause disease in humans, and that expresses the spike protein. The advantage of this is that viruses infect our cells very efficiently. When they infect our cells, they will produce the spike protein. So basically, it's a very efficient system to produce your protein of interest in large quantities in the cells. The disadvantages is, although these viral vectors usually don't cause diseases in human, in some rare cases, some people can elicit an immune response to the virus itself, but this is typically very, very rare. And the last two platforms, the DNA and RNA, I didn't give any example because they are new. And we're going to talk a little bit more about RNA vaccines a bit later, but basically the idea behind these two platforms is just to insert into our body the DNA sequence or the RNA sequence that codes for the spike protein. And we're going to elaborate about that in the next couple of slides. So basically, as you can see here on the left, all the platforms that I talked about, almost all the platforms that I talked about in the previous slide, have been used to create vaccines for SARS-CoV-2. And you can see here, this is the preclinical phase, phase 1, phase 2, and phase 3 of the clinical trials. And those are the licensed vaccines. But because I took this out of a review that was published a month ago, it's no longer relevant because these things change so fast. So I put here, you can see that to the phase 3, which is a later stage in vaccine production. I'll talk about it in 1 second. Most of the vaccine that arrived to this stage are viral inactivated vaccines, mostly in China, viral vector vaccines that you can see here in green, and the RNA vaccines that we'll speak about a little bit more a bit later. Because this is no longer relevant because it's a month old, I'll put here an updated list of the number of the various vaccines that are in the different stages of the clinical trials right now that I took off the New York Times vaccine tracker. Basically, there are 43 vaccines in phase 1. Phase 1 is the phase that you take more or less 100 people and you test the vaccine safety and dosage. We have 21 vaccines in phase 2. In phase 2, after you prove that the vaccine was safe and you have an idea of the dosage you want to use, you follow up the patients a little bit and then you start a phase 2 trial. In the phase 2 trial, you basically expand the safety trials into a larger number of people, a couple of hundreds usually. Then you move into phase 3. We have 20 vaccines in phase 3. Phase 3 are actually the efficiency trials. We didn't test efficiency until now in actually preventing disease or preventing infection. We'll talk about the difference between these two in a couple of minutes too. But phase 3 trials are large-scale efficiency tests. The Pfizer test was 40,000 people. The Moderna test was 30,000 people. And we have 8 vaccines now in early or limited use around the world, and 2 vaccines approved for full use. So the first question people ask when we talk about these vaccines, or the new SARS-CoV-2 vaccines, is how were these vaccines developed so fast? And the conclusion most people reach is, oh, they're not safe, they did everything quickly, I'm not gonna get this vaccine. So I want to show you what happened and that these vaccines are truly safe. They didn't skip any step. And I want to try and show you that nothing was missed here. It was a combination of other things that made it possible. So let's look at what happens when we try to develop a traditional vaccine. So basically, first thing you do is you go into the lab and you try to find a suitable platform, and usually do experiment in animals, or this is called the preclinical stage. You figure out what you want to do, and if your vaccine works in animal, you submit your paper to the regulatory review, and they approve, and they say, okay, you can go into clinical trials. You start the phase 1, which we said is dosage and safety on a small number of people. You have to follow up these people for a few months. And then when that is done, you start recruiting people for the phase 2, which is a couple of hundreds people. And then you need to recruit people to phase 3, which is around 30,000, 40,000 people. And basically, first of all, most of the companies don't really want to invest in vaccines. They prefer to invest in drugs, which are more profitable. Vaccines prevent diseases. So most of the big companies don't want to do that. The second thing is that when you try to recruit patients to these vaccine trials, they usually don't want to come. And the other problem is that for most viruses, we don't have a lot of the disease going around. We had a Zika outbreak a couple of years ago. Now we have very little cases of Zika. So if you want to do a vaccine trial for Zika, you have to find patients that can actually get the disease if you want to prove the vaccine efficiency. After you finish these trials, you submit your papers again to the regulatory agencies, and then you start to produce, and typically that takes 15 years. In the case of the SARS-CoV-2 vaccines, so there was a large financial investments for many governments around the world. There was a high motivation to volunteer. We had tons of COVID-19 patients. So everything related to that went really, really fast. We had the pre-existing platforms from the SARS-CoV and MERS-CoV, from the previous coronavirus family viruses outbreaks that we had before. So that's another thing that made things go faster. Some of the trials were performed in parallel. So during the time when phase 1 was finished, they saw that it was the vaccine was safe. And then usually there's a period that you follow up the patients to make sure that everything is okay. Because everything was so urgent, some of the stages in this case were performed in parallel. So during the follow-up period of phase 1, they already started phase 2. And during the follow-up period of phase 2, they already started phase 3. And then another thing that enhanced the development of the vaccine is that most of these companies started production at risk. So if the vaccine wasn't approved, they were about to lose a lot of money. But luckily that didn't happen. And we already have several approved vaccine. And this is why also the regulatory processes were enhanced. So that's why we were able to get the vaccine in something like around 10 months. And now we are all able to get vaccinated. But as you can see, all the stages was performed. There were no shortcuts. So you can rest assured that it's safe. So here in the table, you can see some of the vaccines that were already, that are in late phase studies or already approved under emergency use authorization. And we can see how some of these vaccines compare. The first two are RNA vaccines. We'll talk about it in the next slide. They have the RNA sequence that codes the spike protein. We're giving in 2 doses. The efficiency for the Pfizer vaccine is 95%, and the efficiency for the Moderna vaccine is 94.1. RNA is a very, very unstable molecule, which is why it needs to be kept in a -70 for the Pfizer vaccine and -20 for the Moderna vaccine, which is kind of a problem when you want to vaccinate a whole country or the whole world. It can only stay a relatively short period in a regular fridge, and that complicates things a little bit. In Israel, we managed to overcome it, but it's quite a challenge. The next 2 vaccines are the AstraZeneca vaccine and the Janssen vaccines. Both of them are viral vectors. They're based on adenoviruses, viruses which usually don't cause, strains which don't cause disease in humans. They're also, the AstraZeneca is given in 2 doses. And Janssen vaccine is the only vaccine that had a one-dose arm, which could have been a major advantage, but today they published the results. The one-dose arm worked, but the 2 doses was much, much more efficient. So there goes our hope for that. Looking at vaccinating the whole world, that could have made a difference, but unfortunately that didn't happen. The viral vector vaccines are much cheaper to produce, so that's important. And the other advantage of these vaccines that they can be kept indefinitely, almost for very long periods of time in regular fridges, and that would be a major advantage again when you need to vaccinate such a large amount of people. The AstraZeneca published their efficiency to be around 70%. Janssen still didn't publish their phase 3 results. The last vaccine is the Novavax vaccine. It's a protein subunit vaccine. It's also given in 2 doses. It's at the end of phase 3. When you look at the data, it's one of the most efficient. It gives one of the most efficient immune response compared to the others, but it is still not approved. Okay, so since RNA vaccines are relatively new, let's talk about them a little bit and explain how they work. So as I mentioned before, here you can see the RNA sequence. It's the RNA sequence for the spike protein of the virus of the SARS-CoV-2, and it's encapsulated inside the lipid nanoparticle. We inject this into the muscle, and the lipid nanoparticle, sorry about that, the lipid nanoparticle fuses with the membrane of our cell, and the mRNA sequence goes into our cells, and their cellular organelles called ribozyme translate the RNA into a protein, and that protein is expressed on the cell membrane where it's presented to the immune system, and the immune system produces antibodies against the spike protein. As I mentioned before, RNA molecule is not very stable, and it's degraded within 8 hours. It's not in our cells anymore. That's the first thing I want to mention. The other thing is, you see here the cell nucleus. As I mentioned before, proteins are made in the cytoplasm of the whole cell. So this RNA that we inject goes into the cytoplasm of our cells. It doesn't go into the nucleus. Some people who are against these vaccines said the vaccine could change your genetic material. This is not true because we don't have a mechanism that transports RNA into the nucleus. Also our RNA is produced into the nucleus, but it goes out to the cytoplasm. So the vaccine, which is injected into our body, has no way to enter the nucleus, and it cannot change our genetic material or DNA. So that's basically how RNA vaccines work. And my last slide, there's a big discussion if the vaccines protect us from infection or protect us from developing disease. I want to explain the difference between these two things. Basically, when we are infected by a natural infection by SARS-CoV-2, it goes into our body through the upper respiratory tract and into our lower respiratory tract, where it creates the actual disease. When the pathogen enters through the upper respiratory tract, we develop an immune response against this pathogen in the upper respiratory tract by producing antibodies called IgA. When we inject an antigen into the muscle, the immune response created will mostly be systemic, and it will produce different kind of antibodies called IgG. It will not produce an immune response in the upper respiratory tract. If we have high levels of IgG antibodies, some of them will reach our upper respiratory tract as well. So infection might be reduced, or because some of these IgG antibodies will get to the upper respiratory tract, it won't be as efficient as natural infection or as people who have these IgA antibodies. the best approach to completely prevent infection will be by intranasal vaccination, but unfortunately, right now we don't have any vaccines that are planned using this platform. So the vaccine trials didn't actually test all of the 30 or 40,000 people who participated in the vaccine trial for the presence of the virus in the upper respiratory tract. So the outcome of the experiment was just testing by symptomatic infection. They tested people before the start of the experiment to make sure that they are not infected, and they tested them before getting the second dose. And between those and after the second dose, they relied on people reporting their symptoms. So now we know that those vaccines are efficient in preventing disease. We're not sure if they are also efficient in preventing infection. We will assume that if we have a high systemic antibody response, it will also lower the viral load and it will prevent people from infecting other people. So Moderna did test some of a subset of the population from their experiment, and they have preliminary result that their vaccine reduces infection by 66%. We also have some new data from Israel. One of the healthcare providers giving the vaccine did some exam, and they also have some preliminary result that the Pfizer vaccine also reduces infections by about 30%, but this is only preliminary results. The number might change with time. So we are pretty sure that we know that the vaccine prevents disease, and we have some encouraging preliminary evidence that the vaccine also can at least reduce infection. So I'll stop now, so Dr. Stern will have time to talk too, and that we will have some more time to answer your questions.
Adi? Thank you for that brilliant overview about the vaccines. So Adi, let's move quickly over to you so we can hear from you.
Okay. So I'm just making sure that you can all see. Is that okay?
Yes, great.
Okay. So I'm going to be talking about the mutations going on in this pandemic. And so luckily Ella has given me a wonderful introduction. And so I'll be able to slightly run through some of my more introductory slides. And so when we speak about corona, just making sure that we're on the same page, the disease is COVID-19, but the virus is actually called SARS coronavirus 2. And it's part of, as its name implies, part of the corona family of viruses. And so how do we know that it's a coronavirus? And the answer is that that very early on, scientists performed genome sequencing. So already a few days into January, just a few days after this virus, after the disease was actually described, the genome of the virus was sequenced, okay? So I'm showing DNA here, and as you can see, there are these letters, right? So the DNA is composed of letters. And that's what it means to sequence a virus. It means to literally read these letters, and these letters have meaning, and we can try and also guess what they mean for a virus. So genome sequencing most often is DNA. So if we sequence our own DNA, it would be DNA, our own genomes, it would be DNA. But actually for many viruses, as Ella mentioned, the genetic material is not DNA, it's RNA, including the coronavirus, okay? And so we knew it was a coronavirus because as this virus was sequenced early on in January, researchers compared its sequence, all these letters, and saw what is it similar to? And it turned out that it's very, very similar to other coronaviruses. And so you see here these other coronaviruses that we know of. It turned out that it was most similar to a bat coronavirus, and that's why currently the hypothesis is that this is a virus which jumped from bats into humans. It's also similar to another coronavirus from a pangolin, which is a form of, it's a type of mammal. It's a really kind of strange conus looking animal. And it's also similar to SARS coronavirus 1, which afflicted humanity in 2002, 2003, and to MERS, and to also the common cold coronavirus. It's 50% identical. The length of the genome is 30,000 bases, and that's very short. Okay, so our genome is 3 billion bases, and so you can understand that this is much shorter than our own genome. But despite it being a very, very short genome, as you realize, the virus can cause a lot of devastation just with this very, very short genome. And so this is another figure, which again, luckily Ella kind of explained. And so we see the genome here, sort of the RNA genome inside the virus and the exterior part of the virus, which also includes the spike protein. And the spike is really under a lot of attention now for two reasons. One, as Ella mentioned, it's what allows the virus to enter the cells. And second of all, it's also what elicits an immune response. Okay, that's what our immune system or antibodies recognize. And Ella has really discussed this very much in depth. So now let's talk about mutations. What are mutations? So we have a genome, and as I mentioned, a genome is basically a long string of letters. They're not really letters, of course. They're chemical bases, and we note these chemical bases with letters. And so there are 4 types of letters: T, A, C, and G. And so we have this original sequence. Let's say that this is the virus. And now what happens is that the virus has to copy its RNA in order to produce new viruses. And when it copies, what might happen is that there might be a mistake in copying. Okay. When this mistake happens, you see originally we had a T here, but now we get a C. Okay. And that mistake is called a mutation. Mutations. And so mutations are generating a lot of interest today in the public. And actually this came out very early on when coronavirus was just emerging. And what it says here is we shouldn't worry when a virus mutates during disease outbreaks. And this is kind of just a point of reference because mutations are a natural part of the life cycle of viruses. There are mutations all the time in viruses. And in fact, every second transmission event, so right, we have a virus that's transmitted to one person, and the second time it's transmission, we get a mutation. Which means, as you realize, given the number of transmission events that have occurred around the world, that there are simply many, many thousands of mutations that have already occurred in this virus. The vast majority of mutations that we observe are actually meaningless from the point of view of the virus. They don't do anything to the virus. Okay, and so this is putting everything in context because we're hearing a lot about mutations, but it's very important to be very careful when we discuss this issue of mutations because really a lot of times they are simply meaningless. Nevertheless, unfortunately, very rarely something happens which is called an adaptive mutation. An adaptive mutation is a mutation that makes the virus better at making more viruses. Okay, and so this is a very important point. A mutation is not likely going to be more virulent on its own. It's not going to create a virus that is more aggressive, not likely more aggressive. And this figure actually illustrates this very well. If we have a virus that really, that kills whoever it infects very, very quickly, then what will happen to that virus is that the virus will die together with the person it infects. Okay, it won't be able to transmit onwards. On the other hand, if we have a virus that most often, just causes us to sneeze and wander around without a mask, then that virus will be transmitted more often. And so that's why in general, when we speak about mutations that are adapted to viruses, it's usually viruses that are either more transmissible, okay, just simply are very efficient at moving on to the next host, or for some reason more likely to survive in our body. For example, viruses that are able to evade our immune responses. Okay, so we have these very rarely adaptive mutations, but of course, when you let a virus spread, like coronavirus is spreading, this very, very rare event actually happens. And so let's talk about what we think is going on now with these rare adaptive mutations. And so here we have the different flags, and I'm sure some of you have already heard about the so-called British mutation or South African mutation, or there's the Japanese and the Brazilian mutation. There's like a bunch of mutations going on. And I'd actually like to emphasize something very, very important at this stage, because I've actually heard people kind of pointing this blaming finger and saying, hey, why, what happened in England or in the UK? Why did they create a bad mutation that is now affecting the world? And this is, of course, not true. I mean, the mutations happen everywhere. And it's actually very, very likely that the British were simply the first to detect what's called the British mutation, because they are the ones that perform the most genetic sequencing in the world by far, more than any other country in the world. Okay, so we have these different British and so on and so forth mutations, and I'd like to kind of elaborate a little bit more about who they are and what they mean. And so this is a list, and we're not going to go through them. But you can see that there's this list of mutations, and so actually it's not one mutation. There are many mutations in the so-called South African strain or South African variant. There are many mutations, and also in the British strain there are also many mutations. And again, this is not in itself anything that causes alarm normally, because again, as I said, we get mutations all the time, and they accumulate one on top of another. But what happened here that caused alarm is that you'll notice that there's a big proportion of mutations in the spike protein. Okay, and so the spike protein again is what is kind of the most important protein in the virus, again from our point of view, and that was a concern here, that it's as if too many mutations had occurred, and they also occurred literally at once in a very, very short period of time. We suddenly saw boom, this entire chunk of mutations appear. And this caused some alarm. This also caused alarm because in parallel, there have been many experiments in the past year that have been attempting to characterize different mutations and what they mean. And so researchers grow viruses in the lab and create different mutations and test what they mean. And this is very, very important and critical research. And there have been already some flags raised following this research about these particular mutations that are present in the South African strain and in the British strain. In fact, one mutation that has already been detected is what's marked here in red. It's a mutation at position 501. It's present both in the British strain and in the South African strain. And it's already been flagged because it seems that, that this mutation really causes changes, as we'll now discuss in 1 second. So what do we know? What do we know about these mutations? So first of all, it seems that the spike mutation 501 that I just mentioned, that is present in both British and South African strains, probably leads to a more transmissible form of virus. It's not more virulent, okay, it doesn't cause higher mortality, it doesn't cause more disease, which is good news and makes sense based on what we know, as I just described, about how viruses evolve. But it does lead to a probably much more transmissible form. This is bad news in the sense that if we, I think all of us feel the burden of walking around with masks and social distancing and really being careful and avoiding meeting people and so on and so forth. And what this means is that given such a variant, we have to be even more careful. We have to add layers of social distancing, and I think this is really hard at this stage to think of how we can do even more, but we do have to. The second mutation is called spike mutation 484. It's present in both the South African and in some of the Brazilian and in one of the Japanese strains. And here, what has been shown again in the lab is that this mutation, viruses bearing this mutation may evade antibodies more efficiently. So here it's also important to note that it's not that a virus with this mutation completely evades antibody, which would really be alarming, but it has a lower response to antibodies. Okay, and so that's kind of why there is this alarm. As I mentioned here with this disclaimer, everything that I'm telling you here about these mutations is based on research that has been performed in a matter of days, weeks, and months. In science, we really are unhappy usually presenting things that are based on a few weeks of research. We like research that is based on a much longer period of time. But in this case, I think that we really had no choice, because these both the British and the South African variant, and all of them, Brazilian and Japan, they all emerged in the probably in the past few weeks or months. And so it's been really vital to generate as much, as a rapid response as possible. And so having said that, I'd actually like to go to my last kind of take-home message, which I think is really important, and I think which is the good news. There is more and more evidence that 2-dose vaccines, which, as Ella mentioned, currently all of the vaccines are 2 doses, will protect from both mutations and will protect from all variants that we know of today. And so I think my most important message to any of you listening here is that it really is super important to obtain a vaccine whenever you can, depending on, again, I know the regulations are different in every country now. The availability is different, but it really is going to be important, because vaccinating not only does it reduce our own probability of being ill, but again, as Ella mentioned, there is more and more evidence showing that it does protect to some extent from infection and from transmission. And when that happens, there's less of a chance that mutations will be created. So the more we reduce this virus and the more we vaccinate, the less there's going to be a reduced probability of me talking about any more new mutations. And with that, I'll end and allow Karen to ask questions.
Thank you, Adi. That was remarkable. What an overview. And thank you for ending on that positive note. I was getting a little nervous there in the middle, but you calmed me down. Okay, we have so many questions, so we might actually even go a little over time with your permission, because there's just so many questions and such a fascinating topic. So a number of questions are coming up are, is it safe if this particular disease, that particular disease, during pregnancy, what stage of pregnancy? And none of us are physicians, and we certainly can't give advice about something along those lines, but is there a good place to get that information? I've been involved now in a study looking at the websites that are coming out of the ministries of health all over the world and actually showing that they're not very good in many places. So how does the average person like me get information to find out? I can't run after my doctor, who probably doesn't know because there isn't that much information out there. Any ideas?
Well, you want to answer, Adi?
It's a tough question. I think that, we're all, I think as scientists, we're all kind of scrambling to get information from many different sources, and it really is very tricky. I personally tend to follow the CDC website carefully, because I think that whenever they do post something, it's very, very cautious and very, very reliable information. But again, they're also scrambling because sometimes there isn't enough, they don't have enough time to update something that came out yesterday, and there are already thousands of questions about it.
Well, I could remind you now that we didn't wear masks for a long time because of the CDC as well.
Yeah, I know. It really is tricky. I think, again, there are also different, the lines of advice are different in every country. For example, regarding pregnancy and so it really depends. And I think that it is important to consult a physician, because I think that every person is also different, right? There's always a combination of being, you have this background disease and this, and so on and so forth. And regulations are different in every country. And so I think that's probably the only way to do it.
And they won't have answers for sure.
On your own. Yeah.
So you mentioned at the end, Adi, about that we're safe from all of these mutations because of the 2 doses. So Ella, do you know what's happening, for example, in the UK, where they were making recommendations for a 2nd dose to come later? Does it put people at higher risk for getting COVID?
So basically, we don't really know. So that's the main problem with this. In the clinical trial where they showed 95% efficiency, they had 2 doses in the Pfizer vaccine in a 21 days difference between them, and we know that gives a 95% efficiency. Every deviation from that might be good or might be bad, but the true thing is that we don't know. In the case of Israel, for example, where we have enough vaccines, we can give them as the protocol says, and then we know everybody's covered for 95%. In other countries, we're in different situations, like England, it's a big question. It's a big question, but because the trial didn't test all of these things. They could be better or they can be worse. So really the FDA and all the other agencies, except in England where they're in a very bad situation, and I don't want to be in their position, to have to make these decisions. But because the protocol, again, is 2 doses and a set time difference, then most of the agency don't want to change from that because there's no scientific evidence or there's no data to support anything else.
I guess what follows from there is, for example, if I had a Pfizer and then they ran out and I had to get now a Moderna or even one of the other kinds with the AAV as opposed to an mRNA, that wouldn't help. I need to get the follow-up of the same type of vaccine, correct?
That would be the very best thing to do because, again, it might be better for what we know, and it might be much worse because it might, generate a hyper-reactive immune response. So until people actually do these experiments, we can't give advice about that because we really don't have the data.
Do you remember, have they been saying which vaccine might be better for people who are over 65 years of age, or do they seem to be as efficient all across the board with the ages that they've checked?
I think as far as I remember, I think I have a slide talking about that somewhere, comparing the Moderna and Pfizer vaccines. Okay, so the Pfizer, the efficiency was comparable across all age groups from 16 to 89. In the Moderna, there's a little bit drop in efficiency. Above 65, it has only 86.4%. But personally, if I was over 65, I would take 86% than risking getting infected with the coronavirus.
So what happens, tell us about, we have a question from one of our attendees. So the RNA has to be, that you talked about, it has to be saved in these really fancy ultra cold freezers. And one of the problems in many places in the world is they don't have access to those freezers. And yet once it goes into our body, it's pretty warm in there. So what happens to the RNA?
The RNA gets degraded. I mentioned that. I showed you the slide regarding the RNA. The RNA, after 8 hours, it's not in our body anymore. So both Pfizer and Moderna did some trick with the RNAs. They changed one of the nucleotide to a nucleotide that is not recognized by our immune system, which is why they're more stable than regular RNA. But after 8 hours, it's not there. But by that time, it already produced enough protein to elicit a strong immune response, which is what happens with these vaccines.
Do you know what's happening with the vaccine in Israel? We'd heard a lot about it in the news for a couple of months, and now it's been a little quiet. Maybe it's not needed as much because of the Pfizer and Moderna and others that are coming out. Or are they just working quietly on it and we're going to find out later?
So they just completed phase 1 and they're recruiting people for the phase 2. That's basically. So we still don't know if we'll need to get vaccinated each year, like Adi said. So I think they're keeping on working on that because we don't know how much we'll need, when we'll need more vaccines. So I think they're still pursuing it. They're just finished phase 1.
Right, so that's a recurrent question. Will we need it next year and the year after, like the flu shot that we get every year?
That's a question for Adi.
Right, and then if I got the Pfizer this year, okay, you can forget the question about the first and the second, but next year, if Kupat Cholim only has the Moderna, will that be okay? Or a completely different kind that's not mRNA based?
Yeah, so unfortunately, I think my guess is that yes, we might be in a situation where we will need the vaccine every year or two. Again, based on what we know from the flu, this virus also has the potential to sort of to evolve rapidly and to change. And so yes, it is possible, but I think the good news is that it's very easy to create this vaccine, and I think we've made a tremendous advance, as opposed to flu vaccines, where the protection is actually not nearly anywhere as high as it is from what we see from these clinical trials. I mean, we're talking about 80-90% protection, and in flu it's much lower. And so I think that will make our personal safety much higher. If we get vaccinated every year. I wouldn't guess every year, probably maybe every second year. But again, this is only a guess at this stage because there are a lot of features of this virus which are new to the entire scientific community.
So you talked earlier about the intranasal vaccinations and the advantage of that. Why has anybody been working on that kind of vaccination for COVID, for SARS-2?
So those are usually attenuated vaccine. And as I mentioned in the beginning, attenuated vaccines are more difficult to produce because you have to prove that they're safe and they cannot get back to virulence. Like what was very rare, but like what happened with the polio. And that's why they take more time to be produced. And that's why we needed these vaccine really, really quick. So I don't think anybody really bothered with that, but maybe I'm not aware of an attempt.
What's the prospect for vaccines for children?
So Pfizer initiated a trial 12 to 16. I don't think anybody tried under that. Typically, those trials take more time because parents usually don't volunteer their children to these things. So, I mean, yesterday I spoke to the Pfizer vaccine, to the guy, the guy responsible for the vaccines in Israel. He said he doesn't know when the trial would end. So I'm guessing it's going to take a couple of months. And I don't think any company is doing trials under 12 right now. But the trial has been initiated in Pfizer for 12 to 16. That's all I know.
Is there any evidence or any updates about whether the vaccine could hurt fertility? It's one of the questions from the participant.
Do you want to answer? You want me to answer?
I think we're probably going to answer the same, right? I mean, so similar to children, there haven't been I'll answer first in the context of pregnancy and women and sort of that side. And so similar to children, usually pregnant women or nursing mothers don't enter clinical trials initially. It's also something which is usually tested first. So the typical sort of way a vaccine is tested is first you test the general public and then you go on to children, similar to what Pfizer is doing, children that are, more adult children, and then only later pregnant women and so on. And so on that front, there, we simply do not have evidence. There actually in some of the trials, in the Moderna and in the Pfizer trials, there were women who became pregnant, and there was no negative indication that anything, there were no problems on that front. Regarding male and female fertility, again, these are these are sometimes studies that you have to wait for years till you realize. But I can see, as a scientist, I can't see any reason why there would be any effect on fertility, because again, we're talking about a very short-lived mRNA. I can't see any reason to assume that there would be such an effect.
There were some claims from anti-vaxxers that there's a short sequence in the spike protein that has some homology to a sequence of a protein that composes the placenta. But according to immunologists, the homology is so low that it's not going to elicit an immune response. And if that was really true, then every person who was sick with COVID-19 who got infected would have a fertility problem, and we don't know anything about that. We don't have any evidence that occurs. So we don't think there should be any issues with that.
One of our participants has heard that people who are immunocompromised might need more than 2 vaccines at this stage. Do you know anything about that?
So based
No, you go ahead.
Okay. So as far as I know, I mean, there is a recommendation for people who are immune compromised to get infected, so to get vaccinated, sorry, so they won't get infected. And I know about a few trials that are running now in Israel where they actually test antibodies in those people. And I think right now they're going by the protocol. All the immunocompromised individuals are getting 2 shots, but we're also, I know that at least 2 trials that are running to test antibodies in these people. And I guess that according to these results, they will decide if to give them further doses or not. There's no recommendation right now because we don't have the data yet.
Well, how often do we get to be in a room like this with experts? Ella, do you have a question for Adi?
I need to think about a difficult one.
Adi, we're gonna turn over the plate to you. You have a question for Ella with her expertise?
Oh yeah, well, we since we teach together, we have so many opportunities to ask each other questions and test each other's knowledge. That at present, no.
So there was a study that actually came out, I think a couple of days ago. I haven't read it myself, but one of our participants asked about it in Science, that the coronavirus is actually going to resemble the common mild childhood cold after the mass vaccination phase is complete. Is there any truth to that? What do you think?
So I think this is based, yeah, this was a very recent publication that tried to really model how, based on what we know on the level of immune response and comparing really, and making a comparison between COVID-19 and between common cold coronaviruses. And so it's a modeling team, and I think that's, you know, they reached these very optimistic results that really that COVID-19 will turn into another strain of the common cold. I think it's too early to tell. I really hope that this is what pans out. There's actually one hypothesis that what's called the Russian flu at the end of the 19th century. There's a hypothesis that it might have been a coronavirus and not flu, and that this Russian flu actually turned into one of the strains of coronaviruses that cause common cold today. And so, you know, if that's true, that's wonderful. But it took 100 years.
They didn't sequence it back then?
They didn't sequence it back then. It's such a shame.
You can probably
I
find
think in the
some.
technological revolutions that we've had that have really allowed us to get to where we are today. It is phenomenal.
Yeah.
So I think I'm going to end with one last question. There were a lot of questions that were similar, and I hope that we got as many questions from the audience answered as possible. But let's say the three of us get vaccinated, and most likely that will happen. I've already had my first dose. But what happens if a large part of the population doesn't, depending on the country or because of some of the people that you mentioned, Ella, the anti-vaxxers? Is that going to compromise me?
I think the first, you know, that we don't really have enough knowledge, again, because we don't know about transmission and infection. So I don't think that we, you know, really answer this very, you know, very rigorously. But I do think that if, and again, this is based on a lot of modeling work that has been done, that if enough people are vaccinated, even with just one dose, we'll be able to bring down this virus to levels where it doesn't really create the problems that it's creating today. I really think it's a matter of numbers. I think the numbers are really important here. Reaching and not getting enough people vaccinated. Again, it should simply depress the numbers of viruses that are circulating wherever you are. And you know, when there's no virus, there's no risk.
Ella, do you want to make one last comment?
By enough, you mean a lot of people, over 70%, right?
Exactly.
Okay, so we're in good shape here in Israel, and we can only hope that we're setting a good example for the rest of the world and how well coordinated the vaccine process has been going on. So what can I say? I really want to thank you so much. I know how busy you are, writing exam questions, teaching, doing your research, and even showing up on TV every couple of days to really help educate the public. And it's so appreciated. It's wonderful to be able to host this webinar from Tel Aviv University, you, Adi, at the Faculty of Life Sciences, you, Ella, at the Faculty of Medicine. And together, I think that we're making just incredible inroads into science and understanding how it's impacting all of us. So I want to again thank all of you, the audience, for joining us. We had quite a record of numbers attending this. It's very relevant for everyone. And I want to thank all of you, and please just be safe and careful and warm. Good night, everybody.
Good night.
Bye.